Hello, can somebody confirm you can hear me?
Can hear you.
Great. Let me share my screen. Are there any questions before we begin? Boy, that's not going to share. There we go. And get out of the way. Gosh. No, we don't need this. All right, today is May 30th, and we're supposed to be on chapter 13, viruses, spheroids, and prions. You'll note there's no lab any day of the week this week. You're supposed to be working on your unknown project. Let me say that needs to be finished by Saturday, June 10th, when you turn in your unknown report. The last day to request a test, and this would be only a test for one day, would be Friday, June 9th. After Friday, June 9th, the unknown notes pages will close. If you do request a test on Friday, June 9th, it has to be a test that you can get back within 24 hours. There's only a few of those. So most of the tests you have to ask by Thursday, June 8th. Any question about any of that? If there's no questions, let me state that some of you have not begun your unknown project. If you do not begin it immediately, you will not be able to finish. As it is, you may not be able to finish. It depends how lucky you are, how quickly you can get the unknown identified. This is really late to begin work on the unknown project. If you're having trouble with the unknown project, message me and I'll try and get you going on the project. If something isn't clear, I'll try and clarify. Like I said, there's a number of students who haven't begun, and you need to begin immediately. This is actually probably too late. You may not be able to finish the project in time. This project does take time to finish. Any questions about any of that? If not, I will be in the lab for the first 15 minutes. I think that's 6:30 to 6:45. Let me look here. Yeah, I'll be in the lab from 6:30 to 6:45 to answer any questions. If you do not have questions, you do not need to show up. All right. Let's begin working on chapter 13. We already began chapter 13, but close that one down. So we were talking about viruses, viroids, and prions, and we had discussed the viral structure of a virus, and that is it has an internal molecule, which is the genome. It's either DNA or RNA, and then that's surrounded by a protein coat that we call a capsid. And some viruses have around that capsid, they have a lipid bilayer that we call the envelope, but only some viruses have an envelope. When we're talking about the general morphology, the shape of viruses, we can classify viruses on their structure, the shape of their capsid. We have the helical viruses, and they are in a helix shape. I don't notice the helix shape in this one here. That looks like it's oh, I forgot the name of that virus. I can't read it there. Ebola, but here I can see the helix. There's also polyhedral shaped viruses, and that would be the many-sided virus that looks sort of like a fancy dice. And there we actually have an actual electron microscopic image of a polyhedral viruses. And then there's a third structure that's just given the name complex. These viruses can be of different shapes and can be of any shape. Here's a complex virus here, and it's, I don't know, not exactly egg shape, flattened egg maybe, I don't know. That one's closer to an egg shape. And then there's one here that looks like the Apollo lunar lander. That's the bacteriophage T4. We'll talk about that. Bacterial virus, that's what we mean by bacteriophage, a bacterial virus, in just a little bit. Any question about any of that? So 3 basic shapes, complex viruses, the helical, helical viruses, and the polyhedral viruses. We're going to talk a little bit about bacteriophage T4. This is the virus that has been studied the most, more than any other virus. And the main reason why T4 has been studied so much is simply that it grows in E. coli. And so it was an easy virus to study. And so people began studying it. And they decided, well, we should really study one virus. And this is the one that they chose to study. It is a virus that infects bacteria cells and specifically infects E. coli cells. Oops, what happened there? It's in the class of the T-even viruses, such as T2, T4, T6. And they all look like this, where they have a head, where the genome is inside of it. You can actually see that there. And the sheath, and the base plate, and the tail fibers. When a virus infects a person, or actually any animal, the virus elicits the host to respond with their immune system. And so it stimulates the host to produce antibodies, and these antibodies recognize and bind to a viral surface protein. And I think we've discussed this before. We call what the host recognizes as foreign, we call those antigens. And all they are is a molecule, usually a protein, but it could be a glycoprotein on a virus, that is on the surface of the virus. It has to be on the surface of the virus because if it's something inside the virus, normally our immune system will never see that, and the antibodies would not bind to it. So it has to be something on the surface of the agent that is infecting us. And we're talking about an infection by a virus. It'll be on the surface of a virus. Some viruses can defend themselves against antibodies on the host immune system by changing or mutating frequently. And so there are different viral strains that can therefore infect the same individual. As you know, COVID-19, you can get that infection more than once, and there are different strains of it. Another good example is the influenza virus. There are different strains of this virus, and even if you get immunized against one strain, another strain can infect you. With influenza virus, it mutates and changes so rapidly that 3 years after getting the vaccine is no longer effective. And in reality, let me see if I can open up Paint here. In reality, your immune system, oh, come on, where's my mouse? So here's our graph. Percent protection on the y-axis and just time on the x-axis. When you get a vaccine, your immunity will be initially high, and it will decrease with time. And once it decreases below a certain level, they tell you the vaccine is no longer protective. In reality, it's slightly protective, but it will decrease eventually to zero over enough time. And after 3 years, it dips below this level where we say that the vaccine against the flu is no longer protective. Any question about any of that? And so that means every 3 years at a minimum, you need to get a flu shot, because after 3 years, the vaccine will no longer be good. And it's just because the virus mutates or changes. And we'll talk about viruses probably on Thursday, that with the flu, it can change dramatically under certain circumstances. And we'll talk about that. In which case, it would decrease to zero as soon as the change happens, in which case you'd need to get a flu shot every year. But like I said, that big change only happens maybe once in about 10 years. This is about on average. And I'll explain to you what it is. It's called antigenic shift. And the virus just changes a lot when antigenic shift comes. And then no one is immune to it. And so you need to get a flu shot that's been designed to fight that changed flu virus. And whenever antigenic shift happens, we'll have a pandemic of flu that year because no one is immune to it. COVID-19 is another virus that changes and mutates fairly rapidly. And that's why probably with COVID-19, you probably need to get a COVID-19 vaccine about once a year. And I'm not sure how long it'll be, but maybe it'll last a little bit longer than 3 years. But like I said, immunity will drop with time because the virus tends to change fairly rapidly, almost as rapidly as the flu, but not quite as rapidly. When we're talking about viruses, we can group them into different groupings, and this is called taxonomy. When we're talking about viruses, they're mainly grouped into families, and then the families are grouped into genuses, and then the genuses are grouped into viral species, and then there are subspecies of one species of virus. A subspecies would be similar to in humans. We have different races of humans. All humans belong to one species, but we have different races. And that's sort of what a subspecies is. There are some viruses which are grouped higher than a family. Let me see if I can remember what that is. King Peter's son came over from, so family, so order. Some viruses have orders, but grouping them higher than an order is very difficult, if not impossible. And the main reason is that viruses don't have very much DNA if they're a DNA virus, or they don't have very much RNA if an RNA virus. And so it's hard to discover the evolutionary history of viruses. And then viruses also evolve very quickly to adapt to their host. And so, like I said, it's just hard going back in time trying to figure out where the virus came from, what viruses are related to each other. There is one exception, and that is we can group viruses into the kingdom that they infect. So there are animal viruses, and they infect animal cells. There are bacterial viruses, and they infect bacteria. There are archaebacteria viruses, and they infect archaebacteria, etc. So we can group them into the kingdom they infect, and that they don't look at the DNA sequence of the virus and determine that. They just look to see what kingdom the virus is infecting. And then we have viruses which are grouped into families. The grouping will be on the type of nucleic acid, whether it's a DNA virus or an RNA virus. We'll take a look at the virus on its replication strategy and on its morphology, the shape of the virus. And that's how we group viruses into families. All viral family names end in viridae, like the family of viruses which we call the herpes viruses. We call that family the herpes viridae viruses. The genus name ends in virus, and so there's like herpes virus type 1, herpes virus type 2, etc. And we generally have a specific name for that. Like there's herpes simplex virus for the genus name. For the viral species, this is a group of viruses sharing the same genetic information and ecological niche. So this would be a virus that infects the same species. Like there's herpes simplex virus, which infects humans. And there's herpes. Let me see if I can remember the name of that one. Chickenpox virus. What the heck is that called? Herpes. I'm just going to call it chickenpox because I can't. Vaccinia? No, that's not right. Anyways, that's the viral species. And all viral species do not have a Latin name. They go by their common English names. So for herpes simplex type 1, that's its name. You can also call that the human herpes virus. And there's smallpox. That would be common name, smallpox virus for the viral species. And then there are subspecies, and they're designated by a number. I'm sure you're all familiar with herpes simplex type 1 and herpes simplex type 2. It's the same viral species, but they are different subspecies. Type 1 tends to infect, doesn't always, but tends to infect the mouse, causes cold sores, mouth and nose, I guess. And then herpes simplex type 2 tends to infect the mucosa of the genitals. The subspecies are very important for viruses. For example, we can say there's the poliovirus type 1, which causes the disease polio, which of course is a very serious disease which paralyzes the patient. And then there's the poliovirus type 2 that we use as a vaccine to prevent patients from getting the disease polio. And the Sabin polio vaccine is actually that subspecies of polio, and we use it as a vaccine because once you get polio type 2, you cannot get polio type 1, subspecies type 1. Any question about that? The important thing is to realize the subspecies and viruses are very important, especially in the terms of the disease they cause. So let's give an example of viral taxonomy. The family is herpes viridae, meaning the herpes viruses of family viruses. The genus would be the simplex virus or herpes simplex virus. It has a common scientific name, but this common scientific name has not been used. People are continually calling it the herpes simplex virus. The common scientific name is the human herpes virus. Like I said, it is not being used in the medical profession or by the general public at large. So I'm not going to talk about it. And then the subspecies would be herpes simplex virus type 1. All right, any questions about viral taxonomy? So we're going to talk about viruses which infect bacteria cells, and these we call bacteriophages. Occasionally, they will be called phages or phages for short. I won't use that term because bacteriophages is just a better term because it tells you that this is a virus which infects a bacteria cell. We'll also later in this lesson talk about animal viruses and then the human diseases that they cause. But initially, we're going to talk about bacteriophages. Viruses must be grown in a living cell, and we can grow them in the lab, a bacteriophage, by simply growing them in bacteria. Here we have an agar plate, and wherever it's white, like here, that's because we have a lawn of bacteria where the bacteria is growing everywhere. Wherever it's dark, that is where the bacteria is not growing. And what we can do with the bacteria on this plate is we can put a solution containing the virus, and then the virus will attach to a bacteria cell, infect the cell, replicate, destroy the cell, and that will release the newly replicated viruses. And then those newly replicated viruses will go to neighboring cells, attach to them, get inside them, replicate, and then destroy those cells. And so with time, we would see that the bacteria will have plaques on it. And wherever we have a plaque, that is where viruses are multiplying. And they'd only be multiplying the outer rim of this plaque, but in the inner rim is where they started. And they lyse the bacteria cells, and that's why the plate is dark there, because there's no bacteria inside the plaque. Any question about any of that? Now, some of these plaques, like this one here, it looks like there's more than one plaque that got together, and that's why that's so big. This one actually looks like it's 3 plaques. Let me blow that up a little more. 3 plaques that got together there. But others of them, you'll notice they're smaller, like these 2 right here. Come on, mouse. These 2 right here are smaller than these right here. And that's probably because these bacteriophages initially bound to a bacteria. And so these plaques began growing at the beginning, whereas these plaques are smaller. And probably what happened was initially the virus was not by a bacteria cell. And what happened was the bacteria started to grow to make the lawn. And then the bacteria eventually grew to the virus. And then the virus was able to attach to the bacteria and then infect the cell and then begin the formation of the plaque. So that's probably why these plaques are smaller than these 2 plaques here. Any question about any of that? Maybe one more. All right. When we're talking about viral multiplication, the only goal of a virus is to infect their host cell and then use it to reproduce. How the virus does that, it actually hijacks the cell. What happens is a virus will bind to the cell, inject itself into the cell, or get inside the cell for human cells, because they don't need to actually inject into an animal cell. And then the virus takes over the host cell's metabolic machinery and runs the cell. And what the virus does is it causes the cell to replicate viral parts to make new viruses. And it uses the host ribosomes, tRNA, amino acids, ATP, and some of the host enzymes to replicate the virus. The basic mechanism for viral multiplication is similar in all viruses. I already mentioned bacteriophages are viruses that infect bacteria cells, and bacteriophages can replicate under two mechanisms. They have the lytic life cycle and the lysogenic life cycle. In the lytic life cycle, this virus will infect a cell, replicate, and then lyse the cell, killing the cell. And that's part of the virus's life cycle. Why they lyse the cell is that's to allow the virus to leave the cell. And that's the lytic life cycle. In the lysogenic life cycle, the host cell remains alive for as long as the virus is in the lysogenic life cycle. Any questions about any of that? In the lysogenic life cycle, the virus and the host cell will continue living together in a form of symbiosis for as long as the lysogenic life cycle continues. So I've got a question for you. To make a new bacteriophage, what do we need? What components does the virus need to replicate in order to make copies of itself? Anyone want to guess? Well, what does a virus have? All viruses have two parts. What are the two parts? You know, if I ask this on a question, you guys, in a quiz, you guys will be in trouble.
The capsid and the genetic material.
Yeah, that's right. They need to replicate the protein capsid, and they need to replicate their genome, which would either be DNA or RNA. Let's take a look at the next slide. So a virus needs to replicate its genome, and here we're just talking about DNA, although it could be RNA, and we'll show you that later. So it needs to replicate the DNA, and then it needs to make the capsid protein. And how it does that, it transcribes the viral DNA to make viral messenger RNA, and then the viral messenger RNA is translated in the capsid proteins, and then the capsid proteins gets together with the viral genome to make the new progeny virions. All right, let's talk about the lytic life cycle of bacteriophages. It follows 5 stages. Trying to open that. Attachment, penetration.
First, the phage particles stick.
Biosynthesis, maturation, and release. It looks like this is not going to play in my other video player. Let's see if I can find it and open it.
First, the phage particles stick to the surface of the bacterial cell. They bind to specific sites called phage receptors. Next, the DNA of the phage is injected into the cell, as if from a syringe. The ends join, forming a circular molecule. Then, the injected DNA replicates inside the host bacterial cell, and phage heads and tails are synthesized. The phage DNA is then packaged into the heads, and the filled heads attached to the tails. Finally, the cell dissolves, or lyses, liberating about 100 newly formed phage particles after 45 to 60 minutes.
Sorry, the video portion of that is not playing, at least in that.
In a way,
there it
a
goes.
virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
Okay. Any questions about that movie? There is one part I wanted to emphasize. Of course, I'm not going to get it. I'm not going to see it again. And that is when the virus infects the cell, it breaks down the cell wall right here. And then the sheath contracts, and that pushes the, puts pressure on the head of the capsid, and that pushes the genome of the virus into the cell. I'll keep this up here because we're going to watch it later for the lysogenic life cycle. This is the movie I have for that one. Like I said, the other one is supposed to have a video, but for some reason on the on this movie player, and I've only got one on this computer, it doesn't play. The video doesn't play, so you only get the sound for that one. All right, where are we? So in the attachment stage of the lytic life cycle, the phage attaches to the host cell. There's then penetration, and what happens is that the lysozyme opens up the cell wall. The lysozyme digests the cell wall, and then the tail sheath actually contracts, and that forces the genome inside the head of the virus into the cell. And let me see if I got, there it is here. So here it is here, and the virus secretes lysozyme, an enzyme that digests the cell wall. Then we have an opening in the cell wall, and then the sheath contracts, and that will push the genome that's inside the head here into the cell, which you can see the genome moving in there. That sort of works the same as putting pressure on a syringe, and that will inject the contents of a syringe into the patient. Biosynthesis is the stage that then follows. It's the production of phage DNA and proteins. So right here, we have the genome going into the cell, and the genome will be copied. So we have different copies of that. And that I don't see in that picture there. Let's see if it's in the next one here. Nope, it's not shown. And then the genome is translated to make messenger RNA. The messenger RNA is translated into viral protein, and we'll get the capsid proteins, the proteins that make the head, the proteins that make the sheath, and the proteins that make the tail fibers. And that'll be followed by, coming up here, maturation. This is the assembly of the phage particles. The capsid proteins all come together to make the protein coat of the virus. And we have an enzyme that's secreted by the virus, meaning the viral genome has this enzyme, and actually stuffs the genome of the virus into the newly replicated virus. And then there's release of the new virions, and that happens by the viral genome expresses the gene of the virus for lysozyme, and the lysozyme will be made by the cell and will go out and digest the cell wall, and that will remove the cell wall so that water can come into the cell, and more water will come in. The cell membrane will expand and expand, and it'll eventually burst. And when it bursts, that will release the newly replicated viruses. And of course, the bursting of the cell membrane will kill the host cell. When we're talking about the lytic life cycle, there's two times or two terms you should know. The burst time, that is the time from when the host cell was first infected with the virus till this time when the host cell bursts, releasing the new viruses. This does depend on the temperature, but for most bacteriophages, it averages 20 to 40 minutes. So it will depend on the species of the bacteriophage, and it will depend on the temperature you're growing the cells at. And then the other term you should know is the burst size. That's the newly replicated virions that are released by the bursting host cell. With most bacteriophages, you don't need to know this number, but it's between 50 and 200 viruses will be released from a bacteria cell. Any questions about any of this? All right, so that is the lytic life cycle of bacteriophages, and there are many bacteriophages that follow the lytic life cycle. There are some bacteriophages that follow the lysogenic life cycle. In the lysogenic life cycle, the virus will attach to the host cell, and we'll get penetration of the viral genome similar to in the lytic life cycle. And then once the lysogenic genome is injected into the host cell, one or two things can happen. The virus can, if it's a lysogenic virus, it can enter the lytic life cycle, or it can enter the lysogenic life cycle. If it enters the lytic life cycle, the virus will replicate both its genome and its capsid proteins. They'll assemble the viruses, and then they'll lyse the cell, releasing the newly replicated virions. And so the lytic life cycle of a lysogenic virus follows the same lytic life cycle that we described above. However, if the virus follows the lysogenic life cycle, then the host cell will remain alive for as long as the virus remains in the lysogenic life cycle. What happens in the lysogenic life cycle is the viral genome, and these are usually DNA viruses, will actually integrate into the host DNA. And so the DNA of the virus will become one with the host cell DNA, and then that will begin the lysogenic life cycle. The phage, the bacteriophage, will remain inactive. It will not replicate unless the host cell replicates. And this host cell we call a lysogenic cell, and it will remain alive for as long as the virus remains in the lysogenic life cycle. Any questions about any of that? All right. So immediately upon entry into the cell by a lysogenic virus, the virus has to decide, am I going to enter the lysogenic life cycle or am I going to enter the lytic life cycle? And this has to be decided immediately once the virus infects the host cell. Like I said, if it enters the lytic life cycle, it will then replicate new virions. It will burst the cell. That's why it's called lytic life cycle, because the cell lyses. And then the newly replicated viruses will go out and infect other bacteria cells. And because it's a lysogenic virus, it will then immediately have to decide, once it infects the host cell, will it enter the lytic life cycle or will it enter the lysogenic life cycle? If the lysogenic virus enters the lysogenic life cycle, the genome of the virus will integrate into the chromosome of the host cell. Let me blow this up a little. So we have the viral genome shown in reddish purple, and then the host cell's DNA and chromosome shown in bluish or dark purple. And the cell will remain this way in the lysogenic life cycle for as long as the lysogenic life cycle continues. The virus will only replicate when the cell replicates in the lysogenic life cycle, meaning the host cell, when it replicates, it'll copy its DNA, and when it does so, it'll copy the viral DNA, and then the cell will divide in two, each getting a copy of the host chromosome as well as the viral genome. And this can go through many cell divisions and continue the lysogenic life cycle. For reasons that are not well understood, and viral geneticists have been studying this for over 100 years, and they still don't have a good explanation. But for some reason, every once in a while, a cell will leave the lysogenic life cycle. And when it does that, the viral genome will excise itself from the host chromosome and then become separate from the host chromosome. And this will then enter the lytic life cycle, where this genome will be copied, the protein capsids will be made, viruses will be assembled, and then the cell will be lysed. Okay? Any question about any of that? All right. As I stated, they don't know most of the time why lysogenic life cycle stops and the cell, or not the cell, but the virus switches to the lytic life cycle. They're not sure exactly what's happening. They do know that one thing that can cause the virus to leave the lysogenic life cycle and enter the lytic life cycle is if you damage the DNA of this cell. If you do, then the virus is likely to leave the lysogenic life cycle and enter the lytic life cycle. And that's because the virus has some way of knowing that the DNA is damaged, and the virus is worried that this cell will die, and so the virus enters the lytic life cycle. But for all other reasons why it happens, they don't really know. I can tell you they do know why the virus enters the lysogenic life cycle and then stays in the lysogenic life cycle. And that is in the lysogenic life cycle, the virus expresses a gene of the virus. So we'll call it this region here. And what that gene is that continues the lysogenic life cycle, it is a repressor protein that shuts down the genes of the lytic life cycle. And as long as that repressor protein is functioning, the cell will remain in the lysogenic life cycle. Any question about any of that? And I guess if that gene were to be turned off, or if it were to not function, the cell will then leave the lysogenic life cycle and then enter the lytic life cycle. All right, let's watch the little video on that. I think I've got it right here.
The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts to new virus particles. Finally, the newphages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions.
So that's the lytic life cycle.
Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle. Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates, and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, any questions about that movie? Let me close that. So when the lysogenic DNA of a virus integrates into the host chromosome, we call that a prophage. And I did mention this, let me should go down. And then the lysogenic life cycle will continue as long as the repressor protein is made, and it will keep the lytic life cycle dormant. And then the cell will replicate normally, even with the prophage in it. There are a couple of consequences of lysogeny. First, a lysogenic cell is immune to infection by the same bacteriophage, meaning that this virus has been inserted into this cell, and that virus will become a prophage in this cell, meaning the viral DNA incorporates with the host cell's DNA. This cell is now immune to infection by this species of virus. That's the first consequence of lysogeny. The second consequences of lysogeny is that the cell can express a new phenotype. We call this phage conversion. This cell here has certain genes with it, and now when we get the viral DNA put into this cell and it incorporates into the host chromosome, this cell now gets new genes, and it's all of the genes on the virus. And that can change the phenotype of this cell. The best example of a change is Corynebacterium diphtherium. Corynebacterium diphtherium produces the toxin, which we call the Corynebacterium diphtherium toxin, only when the bacteria has a lysogenic virus inside it, meaning it's the lysogenic virus that encodes the Corynebacterium diphtherium toxin. So the cell without the lysogenic virus does not produce that toxin. And if it does have that virus in it in a lysogenic cell, then it can produce the Corynebacterium diphtherium toxin. Any question about any of that? All right, that's enough about bacteriophages. Let's go ahead and start talking about viruses that multiply in an animal cell, and these we call animal viruses. Animal viruses surprisingly follow the basic patterns of viral multiplication as in a bacteria virus. But there are a few differences. And this slide is trying to show you that there are differences in attaching to the cell membrane instead of to the cell wall, obviously, because animals don't have a cell wall. There's differences in their entry into the cell. Penetration isn't by the virus injecting its genome into the cell, as happens in a bacteriophage. Animal viruses get into the cell either by endocytosis or by fusion. We'll talk about that. There's differences in the uncoding of the virus. There's a little difference, not much, in the biosynthesis, but the point is you get the same production of nucleic acids and viral proteins in biosynthesis. Maturation is really the same in animal viruses and in bacteriophages. And then the release of the virus can be the same, meaning some animal viruses replicate in a lytic life cycle, the same as in the bacteriophages, where some of them replicate in the lytic life cycle. But we do have some other life cycles, like the budding life cycle, that is only found in animal viruses. The budding life cycle will give the virus an envelope, and we'll talk more about that. There are some differences, like I said, in the multiplication of animal viruses and in bacteria viruses, but the one difference that you must understand is the difference in where the virus multiplies. In bacteriophages, the viruses have to multiply in the cytoplasm because that's the only place the virus can multiply. It's really the only place that there is within a bacteria cell. So they always multiply in the cytoplasm. In animal cells, though, an animal virus doesn't have to multiply in the cytoplasm. They may multiply in the cytoplasm, but they can also multiply in the nucleoplasm. And so that's the main difference you need to know between animal viruses and bacterial viruses. The difference in where they multiply. And I'll talk about this a little bit later, but generally, if an animal virus is a DNA virus, meaning their genome is made up of DNA, then they will multiply in the nucleus. But if the animal virus has an RNA for its genome, in most cases, they replicate in the cytoplasm. There is one exception, and I'll talk about that exception. All right. So there is a little bit of a difference in the attachment of an animal virus compared to a bacterial virus. And the main difference is actually not the attachment, but the way the virus gets inside the cell. Animal viruses can enter either by endocytosis or by fusion. Let me state that there was a mistake in the textbook of the earlier versions of our textbook where it said that animal viruses can enter by penocytosis. That should have been by endocytosis. Okay, so if any of you have the older textbook, just remember that it shouldn't be penocytosis, it's by endocytosis. By endocytosis, the virus will attach to the host cell, and then the host cell will begin invaginating around the virus. And that invagination will become bigger, and then the cell membrane will actually pinch off right here. And so this part of the cell membrane will attach to this part of the cell membrane, and that'll reform the cell membrane. And then this part of the cell membrane here will also pinch off and will circularize around, and that will form an organelle around the virus. And that organelle is now inside the cell, and it'll be brought into the cell. And that is how endocytosis happens. The other way that viruses can, actually, this is more by endocytosis. So let me call up paint again. The other way that animal viruses can enter a cell is, let's make the cell here. Oops, come on. And then our virus here. Come on. I'm not a good artist and it's hard to do that. There's a cell, an envelope on this virus. Come on. Sorry, my mouse isn't behaving. It's not even working. There we go. And of course, there's a double membrane here too. Come on. Well, the envelope of the virus can fuse to the cell membrane. Come on, mouse. And they can then form, let's go like that. Cut that open. Cut that open. There we go. See if I can get my mouse to draw. They can fuse, and then I guess I got it already. Let me think about this. No, that's not right. This should be, I don't have that right. Close that off. There we go. All right, now let's get rid of the eraser. Confused where the envelope of the virus will then fuse with the envelope of the, not the envelope, the cell membrane. And so the cell membrane now will go like this, and then we'll go around the virus, and then we'll continue. So the envelope can fuse with the cell membrane, and now the contents of the virus are then open to the cytoplasm. This is the cytoplasm here, and then that's how the virus can get into the cell. Any question about any of that? Hopefully I kind of have it drawn here. Not a good drawing, but I'm not an artist, so that's as good as I can get. And this is trying to show it here that the envelope of the virus can actually fuse with the cell membrane here. And it's not really shown on this one. This is actually endocytosis here. All right. One difference that animal viruses have is that there are some animal viruses that follow the budding life cycle. So as I mentioned, we do have some animal virus that follow the lytic life cycle, and that lytic life cycle is pretty similar to the lytic life cycle in a bacteriophage. And those viruses will get inside the cell and replicate, and they will lyse the host cell. And when they do that, the viruses, the newly replicated viruses, will be released from the host cell. There are some other animal viruses that follow the budding life cycle. And what happens here is the virus will replicate itself, but not in large numbers. So there will only be a few copies of the virus made. And then we'll put the DNA of the virus, if it's a DNA virus, inside the viral capsid. And then the viral capsid will come up to a membrane of the cell. This can be the plasma membrane, or it can be the nuclear membrane, or in a few viruses, it can be the membrane of the endoplasmic reticulum, or the ER. And then the virus will actually push on that membrane and push further so it invaginates, and then it'll break off a portion of that membrane. And then the membrane will reseal right here. And then the envelope here will reseal. And that is how the virus gets its envelope. The envelope is actually a portion of a membrane of the cell. So if this is the cell membrane here, then this envelope will be a portion of the cell membrane. Any question about any of that? I'm not going to quiz you on this, but the virus does make proteins and glycoproteins, and then we'll insert them in the membrane. And where this virus actually goes and then starts pushing is where these proteins, viral proteins and viral glycoproteins are. And that is how the envelope of a virus will get the viral proteins and glycoproteins. The rest of the cell membrane may not have those viral proteins or glycoproteins. So the virus will be released by budding, and generally the virus replicates at low numbers so that only a few viruses will bud at a specific time. And then the host cell, most of the time, if not all the time, well, it would just say most of the time. Most of the time, the cell that's budding will survive the virus. In fact, if this host cell dies, it is not usually from the virus killing the cell. It is from the host immune system recognizing that this cell is infected with a virus and then attacking this cell and eventually killing it. And how the host immune system recognizes that this cell is infected with a virus is you see these viral proteins and glycoproteins in the membrane. If that's a cell membrane, the immune system can recognize those as being foreign to the host and then can mount an immune response against this cell. Here we have an actual electron microscopic image of several cells. That's a cell here, and there's a cell here, and there's a cell there. And as you can see on this cell here, there are viruses budding out of that cell. And there's one there. Any question about any of that? All right, let me mention just briefly that there is another life cycle, which is similar to the budding life cycle, but it is of a naked virus. What happens with this virus, it replicates generally in low numbers in the cell, the same as the budding virus replicates generally in low numbers. And then the virus, instead of leaving the cell by budding, the virus will actually be incorporated into a vesicle inside the cell, and then the vesicle will come up to the cell membrane and then dump its contents outside the cell. And this we call a persistent viral infection, or that life cycle of a persistent viral infection life cycle virus. And it's similar, like I said, to the budding, except the virus does not bud. And instead of being an envelope virus, it's a naked virus. But it does form a vesicle inside the cell here. And then that vesicle will fuse with the cell membrane and then dump the contents of the vesicle to outside the cell. Any question about any of that? All right. As you know, viruses have different genomes. There's double-stranded DNA genomes, single-stranded DNA genomes, double-stranded RNA genomes, and single-stranded RNA genomes. Therefore, each virus type needs to have a slightly different mechanism for replicating its genome as well as its viral proteins and biosynthesis. And we'll talk a little bit more about that in the next couple of slides. And I don't know if I'll finish that before we run out of time. This slide is showing you the DNA family of animal viruses that cause disease in humans. So you can look over on the column over here. We have the disease that are caused, and then the family of viruses causing the disease. Some of them we talked about before. Let me blow this up a little. Oh, come on. Pox viridae, the pox viruses, obviously smallpox, cowpox. Herpes viridae, the herpes viruses, such as herpes simplex virus. Chickenpox, which isn't shown up here. Oh, there it is. Varicella virus, chickenpox virus, also the shingles virus. And then some other viruses we haven't talked about. I'll talk briefly about Adenoviridae. This is a DNA virus, and it's most famous because it causes respiratory infections that we call the human colds. And there are actually 3 families of viruses that cause the human colds. The adenoviruses are the 3rd family that causes the least human colds. I guess that's the way I'll word it. So this is the family of viruses that cause the fewest number of human colds, but the adenoviruses can cause human colds. Hepatitis B, Adenoviridea, and we're not going to talk about that one. Anyways, you don't need to know the different names of the viruses that cause human diseases. I'm just trying to show you the different families of DNA viruses that can cause human disease. And there are 7 DNA families of viruses that cause diseases in humans. Any question about that? And this table does have some other information which you don't need to know, like whether the strand is double or single, whether the envelope is naked or enveloped, not the envelope, whether the virus is naked or enveloped virus, and then the size of the virus. So for DNA containing viruses, the DNA animal viruses generally replicate in the nucleus. And that's the DNA of this virus. Obviously, the protein capsids are made in the protein capsid is made in the cytoplasm because that's where proteins are made. That's where the ribosomes are. And herpes simplex virus is a DNA containing virus. Once viral DNA enters its host cell, one of 2 things can happen. The viral DNA can recombine with the host cell's chromosome, and that would be similar to a lysogenic virus, but we don't follow the lysogenic life cycle for an animal virus. We don't call it a prophage, we call it a provirus. And the provirus will remain a permanent part of that host cell's DNA for as long as that cell is alive, meaning in animal viruses, if the genome of the virus integrates into the host chromosome, it will never be excised. It will always remain a part of that host cell's chromosome for the lifespan of the cell. And the virus will replicate whenever the host cell's DNA is replicated. The second thing that can happen if the viral DNA enters the host cell, the viral DNA can be expressed. We can get messenger RNA, viral messenger RNA made. We can get viral proteins made. The viral DNA will be replicated. The viral DNA will get together with the capsid proteins. We'll make new progeny virions. And then the progeny virions will leave the cell, either by lysis, if the virus is following the lytic life cycle, or the virus can bud out of the cell if they're following the budding life cycle. So to go through that in more detail, you blow this up. The virus leaves the cell. The virus will attach to its host cell. And once again, there are specific molecules on the surface of this virus, which bind to specific molecules on the host cell. And those molecules on the host cell we call receptors. And the molecules on the virus, we call them either adherins or ligands. Anyways, they attach to the host cell, and then the viral genome will get inside the cell. That can either happen by fusion of the envelope, if this virus has an envelope, or by endocytosis of the virus, and then the virus will be brought into the cell. The viral DNA will be transcribed into viral messenger RNA. The viral messenger RNA will move into the cytoplasm and be translated into viral proteins. The virus in the viral DNA that's in the nucleus, because this is a DNA virus, the DNA goes into the nucleus and of an animal cell and will replicate the viral DNA, and will make copies of the viral DNA. And then the viral DNA will get together with the capsid proteins to make the new virions. And then the new virions will leave the host cell either by lysis of the host cell or by budding out of the membrane of the host cell. Any question about the multiplication of DNA viruses?
It looks like the virions are assembled in a nucleus in the diagram. Is that what happens?
The virus DNA will be replicated in the nucleus.
What about the capsid?
The capsids are made in the cytoplasm. In this case, it does look like the virus itself is being assembled in the nucleus. Okay, that would depend on the species of virus where it is assembled. In this case, it is being assembled in the nucleus, but there are some species of virus where the viruses are assembled in the cytoplasm.
Okay.
Okay, so it just depends on the species of virus. All right, are there any questions? If not, this is a good place to stop. So I think I will stop here, and we'll talk about the RNA animal viruses next time. All right, I'll see you in the lab if you have questions. If not, well, I'll see you on Thursday.